So yesterday we looked at some onion cells, and just a reminder, we had a range of sucrose concentrations. So we had 0.0 molar, which is pure water, and we put some onion epidermal cells in there. So we just peeled off a little layer of the epidermis of red onion, and we let them sit in there for about half an hour. I did it before you guys come into class. We had 0.0, 0.2, 0.4, 0.6, 0.8 and 1.0 molar sugar solution. You then took a microscope slide and you mounted a piece of epidermis and a couple of drops of the solution that it was in, put a cover slip on and looked at it under the microscope. So whenever you looked at it under the microscope, you saw lots and lots of cells. And because they were from red onion, they contain cytoplasm that is red. Okay, so for cells that were in pure water, all you saw was these cells that were just full of red. Okay. When we looked at the field of view for 1.0 molar sugar solution, when we looked at those cells, it was a different story. Because what we saw was that the red colour was only found sort of shriveled up and in the middle. And we reckoned that obviously the 1 molar sugar solution is a really concentrated sugar solution. And that means it has a very negative water potential. And so water would have moved out of the cells by osmosis into the lower water potential of the 1.0 molar sucrose solution. What's the word we use to describe those cells? We say that those cells have been, or they are, plasmolyzed. Whereas 0 moles of sugar is just pure water, those cells are all turgid. The purpose of the experiment was to determine the concentration or the water potential of the onion cells. We want to find the water potential of the solution where these cells are at incipient plasmolysis. So let's do a little bit of thinking. Incipient plasmolysis is when the cell membrane is just touching the cell wall. So if we took those cells there and put them into water, eventually water will start moving in and this will start to get bigger and bigger, and the cell membrane then will just be, there'll be a point where it's just touching the cell wall and no more. It's only touching, so there's no pressure coming from the cell wall. So at incipient plasmolysis, the pressure potential is 0. And if you think back to the equation that you learnt, the water potential of a cell is equal to the sum of the solute potential and the pressure potential. Well, at incipient plasmolysis, that is 0. So at incipient plasmolysis, the water potential of the cell is the same as the solute potential of the cell. And if I left those cells for about half an hour, they will have had time to reach equilibrium with the solution that they were being bathed in. So what we're saying is that if we can find out the cells, if we can detect which cells are at incipient plasmolysis, we know that the solute potential of those cells is the same as the water potential of those cells, which is the same as the water potential of the solution that we put them into. Now, how do you tell? You can't tell just from looking at a slide whether or not the cells are at incipient plasmolysis. It's really hard to tell. And so when we look at these cells, it's the convention we use is that 50%, if half of the cells are plasmolyzed, we use that to determine when incipient plasmolysis is. So what you did yesterday was you counted the number of plasmolyzed cells that you could see, and you counted the total number of cells. And we wrote the numbers on the board. Now, if you've got a good memory, some of you will have realised I've changed a couple of your numbers just to make it a wee bit easier for you. But that's the number of plasmolyzed cells, total number of cells. We could then work out the percentage plasmolysis. So the first thing that you need to do is to produce a results table. Now, what I would do is just get a piece of file paper out and jot this out roughly, okay? So you need a results table. So as I said, obviously, that table I've put on there is rubbish. There's no units. There's plasmolyzed what, total what. Percentage plasmolysis, you might want to put a percentage sign there. How do you work it out? Well, it's the number that are plasmolyzed divided by the total times 100. That's how you work it out. So obviously, for 0.0 molar sucrose, it's going to be 0. For 0.2, well, it's 12 divided by 105. Multiply that by 100. And so it works out at 11.43. Let's get into the habit of using 2 decimal places. So what you will need to do is to, rule this out properly. You can just do it on the proper paper that I've given you there. With a ruler, headings, units where appropriate. And, you don't put the formula in there, but I was just showing you how you work it out. 2 decimal places here in this column here. What's missing from that table then, apart from units and headings and so on? What should every table have? A caption. So we need to mention that it's red onion cells. That needs to be in your caption somewhere. And what is it? It's showing you the number of plasmolyzed cells, number of you need to get into the title that it's plasmolyzed cells, that they're red onion cells, they've been immersed in a range of concentrations of sucrose. Okay, so those are the key things. So that needs to go in your caption. So you need a results table, and then you need to draw a graph. So what are we looking at the effect of? We're looking at here at the effect of the sugar concentration. So that goes along the x-axis. So you've got your sucrose concentration. Okay, so it goes from 0.0 to 1.0, so use a sensible scale. On your vertical axis, you've got percentage plasmolysis, all right? So you're going to work out, and then you're going to plot your figures with a dot and a circle. It might look something like that. In this case, you're going to draw a best fit line, okay? Best fit line is where it is close to as many points as possible. You don't just go from the first to the last, because you're effectively ignoring all the ones in the middle. You put your ruler where it's going to as close to as many points as possible. It may well go through something like that, that's fine. And we want to find out what concentration would give us 50% plasmolysis, because that is incipient plasmolysis. So you need to get a ruler, you need to go to 50% plasmolysis, rule across there, rule down there, and make a note of what that concentration is. So, conclusions. In your conclusions, you need to say that 50% plasmolysis occurred at whatever moles of sucrose, whatever that is there. So we can say that incipient plasmolysis, or we can say at this okay, so you find out what that concentration of sucrose is. We're saying that the cells are at incipient plasmolysis. That means what we've said there, that there's no pressure. You need to say that means that the pressure potential is 0, and therefore you need to explain this. Okay, on the instruction sheet that you got yesterday, there was a table. Okay, so on the instruction sheet you got yesterday, the bottom there's a table which has the relationship between molarity and solute potential of sucrose solution. Now, I've given you that one already. Do you remember when we did the potato experiment? We cut potato into cylinders and then sliced it up. And for part of that, you drew a graph with that information. Do you remember that? Have a look, get out your file, it's probably at home somewhere. If you haven't got a graph, you're going to have to draw a graph for those figures. And so the graph would have the molarity of sucrose solution in moles, and it goes from 0.1 right up to 1. And then it's got solute potential here, but as you can see, the solute potential figures are all negative, so that's why the axes are this way around, because these are all negative, going down to minus 3,500. So when you plot your points, you'll see you get this kind of slight curve. And this is a graph that we can use if we want to find out the solute potential of a particular solution. We read along here. So say it was 0.5. Say you worked out that incipient plasmolysis occurred at 0.5 or 0.45 or 0.33, whatever the figure is there, you go to this graph and you go to that concentration of sucrose solution. Whatever it is, you rule down there, you rule across, and that is the solute potential that causes incipient plasmolysis. These cells that we've been looking at have reached equilibrium. The cells at incipient plasmolysis have the same water potential as the outside solution. The outside solution doesn't have pressure, so the solute potential and its water potential are the same values. So what we're saying is that figure there is the same as the water potential of our cell. The cells are at incipient plasmolysis. So the water potential of the cell is the same as solute potential because pressure is 0. So what we're saying is that figure there that you read off, that calibration graph, tells you the water potential of your cell, which is the same as the solute potential of your cell. And the main aim of this experiment was to measure the average solute potential of cells at incipient plasmolysis. So the instruction says, draw a graph of percentage plasmolysis against sucrose solution. Use the graph to identify the point at which 50% plasmolysis occurs. Actually rule the lines on your graph. Molly. Shh. So whatever X is, whatever that sucrose concentration is that gives you 50% plasmolysis, you go to that concentration here. You rule down there, you rule across. That solute potential is the same as the water potential of your cell. And because there is no pressure, it's the same as the solute potential of your onion cells. So you need to work through all that. You need to describe what you're doing. So your conclusions, you need to say 50% plasmolysis occurred at this molarity of sucrose. That means that there's no pressure because the cells are at incipient plasmolysis. This concentration of sugar has a solute potential of whatever you work it out to be. And because we know that at incipient plasmolysis there is no pressure, we can conclude that the water potential of our cell is the same as the solute potential of the cell, which is in equilibrium with the solute potential of whatever figure you got. Yeah. Are there any questions about that? You don't have an awful lot of writing to do. You're not having to describe trends and things like that. But you do need to go through the process of saying, look, 50% plasmolysis occurred at such and such concentration of sugar. This concentration of sugar has a solute potential of whatever you've worked it out to be. And then you need to explain this bit here about incipient plasmolysis. There not been any pressure. And explain how you work out the final answer. What is the solute potential of your onion cells? And I would like that all done for tomorrow. So you've got 10 minutes now, you've got half an hour. I say don't come here, just do it in the library this afternoon. And then you've got time tonight to spend on it.
If we're not doing the practical, do we still have to do it?
Yeah, you still have to do it because you still need to understand the theory behind it all. Yeah.
What is the solute?
I'm not going to, I probably won't collect it in, but I want to see it, I'm not going to collect it in and mark it if you're not submitting it, but you need to work out, you need to understand all this. So yeah, if you're not actually going to submit your practical, I'll just walk around and make sure you've got graphs and all drawn, okay? All right? Okay?
Yeah.
Any questions?